the issues or the major challenges are really the translation into non-human primates and then into humans. Because I think it's fairly easy to treat a mouse. It's a relatively small brain. It's a not very complex brain. So we have for several neurodevelopmental disorders, we have achieved so far therapeutic benefit in mice. But it really becomes difficult once you go from the fairly simple small brain of the mouse into a larger, more complex brain. And it's not only the size of the brain, which is really provides some struggle in terms of uniform distribution of your vector then, but it's also the increased complexity so that you have a lot more specialized neurons in the primate brain, especially in the human brain, which support the higher brain function. And that's really not something we can easily model in mouse models. Then, of course, the eternal question is, what do you actually need to achieve here? So CDKL5 is expressed, and like many other neurodevelopmental genes, is expressed in virtually all neurons in the human brain. And so it's really hard to predict how many neurons or what particular subclasses of neurons we do have to restore CDKL5 expression, and then at what level really to achieve a lasting benefit in the patient. And as I said, that's really difficult to model in any smaller animal. And I think there's still a lot of thinking has to go into that. To answer those questions, but in the long run, I think once a good product is developed, at some point you really just have to make the decision, go forward and start a clinical trial, because ultimately that only will tell you whether really all of that optimization has played out already, or if a lot more work needs to be done to achieve a therapeutic benefit in patients.